Hello.
Hey, how are you?
Hey, not bad, not bad.
Cool.
Give me a second here.
Mm-hmm.
So what's up, man?
Oh, not much. What's up with you?
Same here, not much. Just been my stomach stuff,
Yeah.
Uh,
What's been going on with that? I mean, what have like the doctors been saying?
what happened?
What have the doctors been saying?
Oh, what do you call it? Not really much. They gave me a new, what do you call it?
Like
Wait,
diet?
hold on. Oh shit. Well, I guess that worked out for me.
What?
It turned out I forgot I had class today for
Mm-hmm.
history, but it turned out he canceled it anyway. So
Okay, that's good. What you said the doctors put you on a new like
yeah, they put me on a
diet?
new called Librax. It's like a half antidepressant too, since IBS apparently has a lot to do with serotonin intake or output.
Uh huh.
So they put me on that
What is it called?
yesterday. Librax, Librax.
Yeah.
No, what do you call it? They're hoping to see if that'll work. But it's going to take a couple of weeks to see any sort of changes
Mm-hmm.
that are noticeable. So,
Oh, it's a benzodiazepine.
yeah. So, that's what it is. Okay. That's the limit. Logged in. Yes. Okay. I'm going to send you over the. All right. I sent you the thing.
Okay.
And then I sent you another thing. This is like how a example test would look.
Okay.
Were you able to get it?
I got the. Oh, okay. Yeah. When is your test? Next test.
And let me get you that date. I think it's October 4th. So I'm just a week and a half out.
Okay. Okay. I can share my screen and. All right, you can see it, right?
Yes. A second. And then there's another one I'm going to send over to you.
Okay. The answers, yeah.
Oh, no, there's another PowerPoint.
Okay.
It's on gene expression.
Okay.
And then here's
Okay, I got it.
Got it?
Yeah.
Okay.
All right, tell me when you're ready.
So I will.
Are you ready?
Yeah, I'm sorry. I'm just having trouble with something over here.
Sure,
One
yeah.
minute. Leave it. Okay, cool. All right. Not that. We're all good. We can start.
Okay. All right, chapter 7, from DNA to protein, how cells read the genome. So, energetic cost of translation. So this is just, the first one is essentially when it binds to the transfer RNA, the tRNA.
Yeah.
And then there's initiation, and then elongation. It's 1 GTP for each amino acid that you add to that peptide chain.
Yeah.
And then termination is when it ends. So when that peptide chain is complete.
Nice.
Okay. So this is just defining like a gene. It's just a region of DNA that's transcribed.
Mhm.
So inside of the nucleus, the gene is the part of the DNA that gets transcribed. It first is transcribed into pre mRNA,
Okay.
and then it gets turned into actual mRNA. So do you remember what the difference was between mRNA and what was originally transcribed?
Say that one more time.
Oh, the difference between like RNA that's initially transcribed and then it turning into mRNA.
Oh, RNA that was previously transcribed turning into mRNA.
Mm-hmm.
Yeah, I believe so. Yeah.
Yeah, there are three things. So one is the splicing out of getting the introns out and just keeping the exons.
Okay.
Remember, like alternative splicing?
Mm-hmm.
Yeah, because when
Yeah, alternative splicing. That's right. Yes.
Mhm.
Sorry. All right. Sorry. My mind goes.
yeah.
No.
Because yeah, first like the gene part of the DNA, it's transcribed like exactly, and then it undergoes alternative splicing to take the introns out because that's the part of the gene that isn't translated. So it just keeps the exons, and and then you add the 5 cap and the poly A tail. So those are the three things. So like in between, like it going from pre mRNA to mRNA, it undergoes the three things, and it's alternative splicing, it's addition of the 5 cap and the poly A tail. The last two things, the 5 cap and the poly A tail, just protect the mRNA from being like digested in the cytoplasm.
Mm-hmm.
So they wrote mRNA allows the cell to separate information storage from information utilization. So like essentially like inside of the nucleus, it's just the storage of information, and the DNA is transcribed into the pre-mRNA, and then when it turns into mRNA, it can leave the nucleus and utilize that information by translating it into a protein. So they describe it pretty nicely here by saying transcription is copying it in the same language, and translation, it goes from the language of nucleic acid to the language of protein.
Okay.
Okay, and now so this slide, this slide over here says that genes can be expressed with different efficiencies, and that's kind of shown here with gene A. It's transcribed into RNA and then translated. So each of these like strips of RNA gets translated. So it's a higher efficiency, meaning that it's going to translate more of that into protein. Whereas gene B, it's less efficient. Like it's making just one copy of the RNA and then just one protein from it.
Okay. Wait, can you explain that one again?
This one?
Yeah.
Oh yeah, just so it says that genes can be expressed with different efficiencies. So here we have gene A. It's expressed with a higher efficiency than gene B because it transcribes like several copies, like 5 copies of the RNA, which makes 5 different.
Which means the
Well,
protein is bigger.
yeah,
Yeah, it makes 5 different proteins.
yeah. Well, like 5 of the same protein, but yeah. And gene B, it's just expressed less, so it just makes like 1 copy of the RNA and then 1 copy of the protein.
Mm-hmm.
Yeah.
All right. Next.
Mm-hmm. So now RNA has a complex 3D shape, and just, this is true for like essentially everything that we're going to do in like biochem and cell biology. The shape pretty much dictates its function.
Mm-hmm.
Like form, basically, like the form of it, the shape of it, basically tells you what it's going to do, or tells it what it's going to do. And its folding is driven by complementary base pairs. So the RNA is, it's still single-stranded, but there's going to be regions where it will bind to itself. So there's going to be double-stranded regions of the RNA. So in this image, you can kind of see that. So in this image, you still have the single-stranded RNA, but it can bind to complementary regions on that same strand to create regions that are double-stranded.
Okay. 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 So next.
Okay.
Actually,
Yep.
wait. What's the difference between the two again?
So this is just saying that RNA has a complex 3-dimensional shape, and so see how we know RNA is a single strand, but it can it can form base pairs with itself to create these regions that are double-stranded, like here and here and here and here. So that's where...
Oh, it can create base pairs within themselves to create a double strand?
Yeah, double-stranded regions. Mm-hmm. And
Okay.
so that just contributes to its overall, like, complex 3-dimensional shape. So it looks like, kind of like this in C.
Okay. What you're drawing there?
Oh, I haven't drawn anything yet, but if I do, I'll draw it here.
Oh, okay.
So, like, maybe I would write, like, Rna forms, or rather, single-stranded Rna can single-stranded Rna has bases that can single-stranded RNA can bind to itself to create double-stranded RNA regions, contributing to a complex three-dimensional shape. And the shape dictates its function. Oh, let me zoom out here.
So the shape of the RNA dictates its function?
Yeah, and you'll see this with a lot of things in like biochem and cell bio, where the shape will always dictate the function.
Okay.
So here's like some single-stranded RNA, and these are the double-stranded regions. Like I made like the base pairs blue.
Yeah.
Yeah. So it's going to have like a specific shape, and this is going to be relevant because we're gonna look at like transfer RNA, tRNA, which has like this type of like clover type of shape. It'll kind of look like this or something. Like tRNA will look kind of like this.
Yeah, the tRNA.
Mm-hmm.
That's the transfer RNA.
Mm-hmm. And then this is just so inside the nucleus, it's information storage. And in the cytosol, it'll be information utilization. So DNA undergoes transcription to produce Pre Mrna, which then undergoes those three things: alternative splicing, addition of a 5 cap, and a poly A tail to turn into mRNA. And then mRNA can leave the nucleus and enter the cytosol, where it'll have information utilization. So that's kind of like the flow of information in a cell. The mRNA is the thing that separates the information storage from the information utilization. Utilization just means using it, so it uses the information to turn it into like a protein.
I see. Okay.
Yeah, and you see that in this drawing over here, right? Like, essentially you have the DNA here, it's transcribed into this RNA transcript, and then in this arrow you see how there's exon here, and poly A tail, so, it basically gets rid of the introns, which are these light pink areas over here. So this drawing of this is the pre-mRNA, and then this over here is the mRNA. So the exons are the darker pink regions, and the light pink regions are the introns. So the introns are gone. So that's why in this mRNA it's just the darker pink, right? Mm-hmm. And then, there's the 5 cap over there, and the poly A tail as well. So those are these three things that are done to make pre-mRNA into mRNA. And whatever I'm drawing here, I can just send to you after.
Okay.
Yeah.
I've been drawing some as we go, but yes, you can still send it.
Yeah, and this is just some more. So in order for something to go from the nucleus into the cytoplasm or vice versa, it goes through these nuclear pores. So that's what that is over here. There's one there. This is a different angle of it. So yeah, it's very, very specific. The movement of things from the nucleus to outside of the nucleus or vice versa, it would need a very specific nuclear signal code or something like that that'll enable it to do that. So yeah, the mRNA essentially, once it's out in the cytoplasm, it will get these ribosomal subunits, like the large subunit and the small one will kind of clamp down on it like this. Mm-hmm. And then we'll have the translation part, which they'll go into in the later slides. Okay. Genetic info directs the synthesis of proteins. So this is exactly just more along the lines of what we've been talking about. So yeah, transcription. So this is they're just going to go over the basic process. So the DNA template strand provides information for the synthesis of an RNA strand. Then there's there's mRNA, which is the code for that's going to be translated. There's tRNA or transfer RNA, which helps it get attached to the ribosome. And there's rRNA, which is just ribosomal RNA, which a ribosome is essentially made of.
Okay.
So essentially you have, DNA, which then can turn into these three types of RNA. So the transfer RNA, and you can see in this image of it, how, so like the mRNA is going to be just one, single strand over there. And
Yeah.
for the transfer RNA, you see how it's like, it has this kind of complex three-dimensional shape here because it's forming base pairs with itself to make that type of shape. And the ribosomal RNA will be kind of like that too, except not as complex.
Okay.
Are you ready for the next slide?
Yeah.
Okay. So now we're going to talk about the difference between the coding strand and the template strand. So for just, double-stranded DNA, right?
Yeah.
So let's say this top one over here is the coding strand. That's going to be the strand that has the code for whichever thing that we're translating or transcribing. And then the strand that's complementary to that, we call the template strand. And since it's complementary to the coding strand, the template strand is what the mRNA, or rather the RNA, uses when it synthesizes the RNA. So here, I'll basically show you. So like, so here's the double-stranded DNA over here, and it first thing it does is get unraveled, or like it separates from the double strand into the two separate strands.
Yeah.
So this one is the coding strand, and this is the template strand. And since they're complementary to one another, and since RNA is going to be complementary also, that is the one. So essentially, I'll use like red, I suppose. So the RNA that gets synthesized from, the template strand is going to produce, just because the template strand is complementary to the coding strand, the RNA that's made from the template strand is going to be identical to the coding strand.
Okay.
Yeah, so the RNA that's synthesized from the template strand is going to be identical to the code of the coding strand.
Okay.
And so both DNA strands are not used to make the RNA. So also these like have different names as well. Like the coding strand is also going to be called the sense strand, and the template strand is the template strand is going to be called the antisense.
Mm-hmm.
And the mRNA is just going to be made from the template strand, but the coding strand, that's not going to make the mRNA. That's just going to essentially bind back to like DNA, essentially, to keep that going. Mm-hmm. So essentially we have, here's a drawing of the polymerase movement. So, you have the unwinding of the DNA here, and it creates, this open region where, the coding strand is up here and the template strand is down here. And, the RNA is synthesized from the template strand. So, it's going to be added to the 3' end. So that's why I kind of drew it like this with this arrow over here. But it's not going to be using the coding strand.
Got it. Okay.
Yeah. So, and here's like an example. So like, here's the coding sense strand, 5'3", it's ATG, GAA, blah, 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 blah. And then the one that's complementary to that is down here. And these will unwind. And this mRNA that's going to be in the 5'3" direction is going to be made from the template strand, and it will be identical to that of the coding strand, just in RNA. Yeah, and then I wrote here, this is the only DNA strand that's used to make the mRNA, the template strand.
Only DNA strands you make RNA is a template strand?
Correct.
Okay.
Okay. All right, now we have. Okay, these are just essentially examples of, so like the antisense is the template strand, sense is the non-template strand or the coding strand, and the mRNA is going to be identical to that sense strand, but just, in RNA instead of DNA. And then each of these, like a codon, is just, three like nucleic bases that code for a specific amino acid. So like GUU codes for valine, and etc.
So with that drawing that you made on the bottom there with the coding and template, when they split apart, right? Can you explain that part again?
Yeah, like this is like like here they split apart here, right?
Mm-hmm.
And then the RNA polymerase works on the template strand to create that RNA strand that is going to be complementary to the template strand. To create one, since the template strand is complementary to the coding strand, the template strand is what is used to synthesize the RNA that's going to be for the coding part of the gene, for the part of the gene that's actually going to be translated.
Okay. So the one on the left is what's responsible for. Yeah, that one.
Mm-hmm. Yeah.
It's responsible for the RNA synthesis?
Yeah. That the RNA synthesis is going to be done using the template strand as a template.
Mm-hmm. And then what does the coding strand do?
So it's going to. So when it uses the template strand as a template, that RNA that's going to be synthesized is going to be identical to that of the coding strand. And the coding strand is just going to, it's not going to work towards making any RNA. It's just going to basically like base pair up with DNA to kind of close that that like this bubble over here. So once it gets rewinded, so like when it gets unwound, that's when we separate the strands and we give them space to be transcribed. And then when that's done, it gets rewound. So this is kind of like different from replication, because in replication you have both strands replicated, like copied, but in transcription it's only one of the strands that's transcribed.
Okay. 1 second.
Yeah.
Hello? Huh? Okay. Okay. Alright, I'm back.
Mm-hmm. Yeah, here. I'll draw like another diagram here.
And what'd you write in the blue there?
Oh, so this is the template strand is the only DNA strand that's used to make mRNA.
Okay. Okay. I'm caught up.
Yeah, and so here's like another kind of like drawing. So like, essentially, here's the sense strand on top, and the antisense strand on the bottom. And so the RNA polymerase is going to bind to the antisense strand, and it's going to start making the RNA. So that's what I drew there.
Okay. That's another helpful one. Okay.
Mm-hmm.
Sorry, I'm taking a while to like understand it. It's just a little.
Oh yeah,
confusing.
no, it's okay. Yeah, there's going to be some additional stuff for my notes that I can add here.
Okay.
Like essentially, why is RNA unstable and things like that. So, apologies for the handwriting.
The RNA binds.
Wait, what?
Oh, sorry.
Because
the RNA binds to the antisense strand.
uh-huh.
The RNA polymerase binds to the antisense strand in order to create the RNA strand.
Yeah, and since it's going to be complementary to the antisense strand, it's just going to be identical to the sense strand, right?
Yeah. Okay.
Yeah, and my pen is still like broken, so the tip moves a lot.
Yeah.
for that.
No, don't worry. It looks fine. I was just asking.
My handwriting's bad, but not that bad. So yeah, this is just some additional information as to why RNA is unstable. And it's because of a couple of things. So like it's 2 prime OH group can nucleophilically attack the phosphate backbone, causing hydrolysis as the remainder of the chain acts as a leaving group. And it's also why it uses U instead of thymine. U is less stable, but it's also less energy expensive.
All right. Got it.
Yeah. So yeah. And when you think of gene expression, just think of transcription.
Got it. Okay.
Okay. Let's see. All right, promoters. So RNA polymerase does not start at random sites on the DNA. It's going to be it's going to start at a specific site, essentially where that promoter is, and then everything. So let's say here is where that promoter is, so that will be the start point, and everything to the right of that, that's towards the 3' end of the gene, is going to be downstream, and everything that's to the left of that, towards the 5' end of the gene, is going to be upstream.
Got it. Okay.
Yeah, and I kind of drew that over here. So like for this, in this diagram over here, like I wrote like here, right here is where it starts. So like, you know, this polymerase, like it already has some RNA that it made, so it started previously. Like let's say it starts right here. So you know, this is like 5-3 direction.
Is this for promoters or?
Oh yeah, we're gonna get to promoter because we're just talking about where it starts.
Oh, okay.
So yeah, so like here, if this is where it starts,
yeah,
everything that
Sorry, I thought I saw something for a second. It's a feather.
Okay. So yeah, so if this is where the transcription starts, things to the right of that are going to be just considered downstream, and things to the left of that are going to be considered upstream.
All right, so transcription starts from where the RNA polymerase is, you're saying?
Oh, so I drew this RNA polymerase over here, and since it already has created this part of this RNA strand, it means that it's already been going for a while. So let's say this is the start site right here.
Yeah.
So this is the start site, yeah, and then to the right of that is downstream, to the left of that is upstream. Here, maybe in purple I'll write something like this. Yeah, so here I just wrote like in that essentially where it starts, right, is where a promoter will be, and that's just a part of the strand of like the DNA strand essentially that is a binding site for the RNA polymerase. So this polymerase that I drew here, it starts by binding here. It binds at the promoter. So that's what a promoter is. It's the binding site for the RNA polymerase, and that also means it's also where transcription starts.
So the RNA polymerase binds at the promoter to create the strand?
To start the transcription.
To start the transcription process?
Yep. Yep. Okay, let's see. Both strands can encode genes, but not in the same spot on the DNA. So that's just saying that the sense strand and the antisense strand is relative to whichever gene that we're talking about. So if we're talking about, so it doesn't mean that the sense strand is always going to be the sense strand for every type of gene. It just depends on which gene that we're talking about.
Okay.
So like for instance here, like for gene A, right?
Okay.
So gene A is located on this top strand, right?
Yeah.
So that means that this top strand is going to the template strand. All right, so sorry. So this strand on the top is the one that has gene A on it, and that means that whatever is complementary to it will be the template strand for gene A. So that will create an RNA transcript from that template strand. So this one down here. But for gene B, the gene B is actually on this bottom strand. So that means that the template strand for gene B is the top one. So the RNA will be made from gene B, like up here.
Okay.
Yeah, so like in other words, let's say that we have so this is, just double-stranded DNA. So if there's a gene that's up here, I'll just say like gene A is up here. The RNA for gene A is going to be made down here, right?
Yeah.
And so if gene B is here, RNA for that is going to be made up here. So essentially this is saying that so here, this up here, I'll do it in purple, is the sense strand, and then down here,
bottom is the antisense.
Yeah, for just gene A. But for gene B, the bottom one is the sense strand, and the top one is the antisense strand. So this is just essentially saying that both strands can encode genes, but not at the same spot. So it isn't like this one is the sense strand for every single gene.
Got it. Okay.
Okay.
Makes sense.
Let's see. Very unusual for eukaryotes if both strands in one area encode for a gene. So, I remember seeing this in your PowerPoints from last time, or like last week or whatever, or 2 weeks ago. But for eukaryotes, you're basically not going to have genes on both strands. So that's why we essentially have so the genes are these like peach colored things. So like for this top strand, we have, genes here, 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 and here. And complementary to that should not have any genes. Because complementary to that is just going to be the template for that gene, right? Mm-hmm.
Okay. Alright, so next.
Yeah, so this is just showing you that, so for any of these genes, complementary to that should not be another gene. Because complementary to any gene, complementary I mean on the other strand, it should just be the template for it, aka not the coding strand, right?
Mm-hmm.
So that's why you don't have any regions where there's a gene on both strands. Like if there's a gene on one strand, opposite that on the other strand is just going to be, it's not going to be a gene because it's just going to be the template for that gene.
Yeah.
So that's why we don't have any overlap like that. Did you freeze? Oh no. I thought you froze or something.
No. Just looking.
Yeah. Because yeah, so like for instance, like gene A down here is the antisense for gene A, and that should just be where it makes the RNA. It should not have gene B like on the opposite end of that.
Alright, so next.
Okay, so the promoter sequence orientation determines which strand is the template, and the polymerase always moves from the 3' direction to the 5' direction along the template strand. So in this like whatever diagram here, for A we have the DNA double helix. So it says an RNA polymerase that moves from left to right makes RNA by using the bottom strand as a template. So it essentially, so the bottom strand is 3-5, right? Like 3' to 5' direction. So the polymerase moves in that 3' to 5' direction. So essentially, like up here, you can kind of see it. So 3' here, 5', and the polymerase is moving, from left to right. It's moving, in the right direction, going this way.
Mm-hmm.
So it's moving in the 3' to 5' direction to create RNA that's complementary to that, aka 5' to 3'. So in this drawing that I drew over here, for this gene, Here, do you see what I'm seeing, or? So
Yeah.
like, yeah, for this gene over here, this circle here is the polymerase, and it's moving to the right to produce this RNA.
Mm-hmm.
Whereas for this gene, for gene B over here, since it's on this strand over here, the polymerase is up here and it's moving to the left. So over here, since for gene A, since the bottom strand is used as a template, it's going to be moving to the right. And for gene B, since the top strand is the template, it's going to be moving to the left. The polymerase is going to be moving to the left, that is. Yeah, and I wrote in red over here, the polymerase always moves from the 3' direction to the 5' direction along the template strand.
Okay.
Okay. Let's see. Overview of Rnp synthesis of RNA. Okay, so there's a DNA-dependent RNA polymerase. That is the copying enzyme. And one of the DNA strands is the template. The promoter is the DNA binding site. And then there are transcription factors, which are proteins that help the phosphorylated the RNA polymerase. It helps the RNA polymerase recognize the promoter. So Rnp is the RNA polymerase.
Okay.
Yeah. So this, big, like yellow oval over here is the RNA polymerase. And, yep, it's basically transcribing that RNA.
Mm-hmm.
And a closer look you kind of have here. So this over here is the DNA template over here. And, it's base pairing with this growing RNA chain. And, it grows in the 5 to 3 direction.
Okay.
Yeah. Yeah, and each time, so like essentially like, because this is the phosphate backbone for that. So this is like what happens like in between. So like, this is like the 3 end of this growing RNA chain, and this is the next RNA like nucleic base. And so the OH group nucleophilically attacks the phosphate group, binding it to that, and then it, that phosphate group, which is essentially just, So, like, you know what, like, a DNTP is?
Yeah. It's DNTP is. I know it. Hold on. So stand for dinucleotide something?
Yeah, triphosphate.
Yeah, dinucleotide triphosphate, yes.
Yeah. So, and the N is just and the N part is just what can be, substituted. So, like, an example of these would be like ATP, GTP. You don't really see TTP or CTP, but essentially that's just, it's adenosine triphosphate, guanine triphosphate, thymine triphosphate, cytosine triphosphate, uridine triphosphate. So, that's why they have, 3 phosphate groups here. Because what you have here is this is an example of a DNTP here. So when it gets added to the growing RNA chain, the the free 3' OH group is the thing that binds to the phosphate of that new DNTP, and it causes one of the phosphates to bind to it, and the other 2 just leave. So that's what we have here. Okay.
Okay.
All right. Let's see. Some differences between DNA replication and transcription. So A turns into T in replication, but A turns into... Well, that doesn't say turn into, sorry. It says A binds to T in replication, but A binds to U in transcription. You know that. Unlike replication, the new RNA strand does not remain hydrogen bonded, but is displaced and the DNA helix reforms. So, so that's why, like, for this one over here, since it's just this one that makes the, the RNA, the other part of it just reforms into DNA.
Okay.
And because RNA is copied from only a limited region of the DNA, the RNA molecules are much shorter.
Oh, okay.
And transcription does not require a primer. So, a primer, if you remember from DNA replication, a primer is just that a primer is just a part of the RNA that has a free 3' OH group, which enables, like, it to be like a DNA polymerase to add more DNTPs to it.
Yeah.
But transcription only works on the side that already has a free 3' OH group, so it doesn't need the primer.
Okay, I see. Right.
So I wrote promoter versus primer. The promoter is a binding site for the RNA polymerase, aka where transcription starts. And the primer is the binding site for DNA polymerase, and that's where replication starts. So, that makes sense?
Yeah.
Okay.
Can you enlarge that really quick so I can write it?
Yeah. Like, wait, which part? Here?
Promoter primer.
Mm-hmm.
All right. Yeah, that's perfect. All right.
Okay, this is just a image of several RNA polymerases on a single DNA strand. So here's like a strand of DNA, and then here's like several RNA polymerases. And so it's a nearly immediate release of RNA from the template allows many RNA copies to be made from a gene in a short time. So that's basically saying that once the RNA polymerase, binds to the promoter and starts transcription, when it ends, the polymerase just immediately gets released, and it lets another polymerase come in and transcribe another copy of that gene. So the immediate release,
all right.
allows many RNA copies to be made from a gene in a short period of time.
Okay. Which basically aids in the expression of one.
Yeah, and the efficiency, if we go back to this over here. So like DNA, several, this is like, 5 RNA strands that are made. So like one strand is made, and then that polymerase like detaches, and then another one comes in, does the same thing. So yeah.
On it.
Alright, now transcription in eukaryotic cells. So, we're going to have different types of RNA here.
Yeah.
So they're just going to go into it. And then these are 3 distinct types of RNA polymerases. Prokaryotes, they just have 1. But for eukaryotes, there are 3.
Okay.
Yeah, and I kind of simplified this over here by saying, RNA polymerases. So polymerase 1 essentially just transcribes rRNA, ribosomal RNA. RNA polymerase 2 transcribes mRNA, and RNA polymerase 3 transcribes tRNA. So that's kind of like what they have here, but just, I kind of like simplified it.
Got it.
And then they say mRNA, tRNA, and rRNA are all made as larger pre-RNAs that are processed by cut and paste reactions. So that's talking about like a transposon, I think. I think they'll talk about it later. And then these are just different types of RNA. So like snRNA is small nuclear RNA. And, it's going to also have proteins that help process mRNAs.
Got it.
And SNO RNAs are small nucleolar. So that's referencing the nucleolus, which is inside of the nucleus. The nucleolus is, it's inside of the nucleus, and it's the thing that makes the ribosomes, or the rRNAs which make the ribosomes, because there's ribosomes inside of the nucleus, right?
Yeah.
Yeah. And then miRNAs are microRNAs. They regulate gene expression.
Okay. Okay.
Okay. Let's see. Transcription unit is a sequence of nucleotide that codes for a single RNA. All pre-RNAs are made as larger forms that are processed into smaller pieces. So that's just basically saying that it's just like a pre-mRNA is a thing that is then is a larger form that is made into a smaller form, which makes the mRNA. So it's just the same thing for the tRNAs and the rRNAs.
Okay.
Yeah. So transcription in eukaryotic cells. So I guess first here they're going to talk about the ribosomal RNAs.
Mm-hmm.
So, okay. So, you know how it says the structure is important for pretty much its function? Like form dictates function. So we pretty much have a picture of it here. And you can see that it's has a very complex three-dimensional shape.
Mm-hmm.
Because, of all these double-stranded regions and stuff.
Yeah.
So, this is a very complex, kind of shape over here. And these are just reasons that the structure is important. So it's important for recognizing other molecules. It's important to provide structural support. And it's also important to catalyze amino acid covalent linkage. So yeah, I would write I guess those three down for ribosomal RNAs. And this last one is important for recognizing that RNAs can also behave as enzymes.
And what did you say about the last one?
Catalyze. That's like so. So I'm writing down some stuff here, and essentially, this last one over here just is showing that the RNA can behave as an enzyme.
Oh, okay.
Yeah, and that's pretty like important just because because mostly all enzymes are proteins, right?
Yeah.
But this is showing that RNA itself can act as an enzyme.
That rRNA itself, right?
What's up?
You said RNA. Is it all RNAs or only rRNA itself?
I would say, this is specifically for our RNA, but in general, yeah, RNA itself can act as an enzyme, just because of the fact that it can create these types of like three-dimensional structures, right? Because for like proteins, right, there's like a primary structure, secondary structure, and once you get to the tertiary structure is the three-dimensional aspect of a protein.
Yeah.
And proteins, like catalysts, or rather enzymes, are usually proteins because proteins have that tertiary three-dimensional structure, which means that they can take a substrate in and turn it into a product. Now, since RNA itself can also form like a tertiary structure, like a three-dimensional structure, it's going to also be able to act like an enzyme.
Got it.
Yeah, so I'm writing some like additional stuff here, like RNA can so it's like it can be a polymer from which ribosomes are constructed. Acts like an enzyme when it's assembled in a ribosome, and it also polymerizes proteins. So polymerizes protein and catalyze amino acid covalent linkages are essentially the same. Yeah. Does it make sense that and polymerizes proteins are the same?
What, that rRNA?
That catalyze amino acid covalent linkages is the same as polymerizing proteins.
Oh, yeah.
Because, yeah, it catalyzes, when one amino acid is binding, so ribosomal RNA will catalyze a covalent
Same as
bond.
catalyzing proteins, you said, right?
What's that?
Same as polymerizing proteins, right?
Yeah, because when you take an amino acid and you make a covalent bond between it and another amino acid, you're essentially taking these amino acid monomers and turning them into a polymer, right?
Mm-hmm.
So that's the polymerizing proteins part.
Fine.
Okay, so now let's see. Okay, so cells have millions of ribosomes. 80% of RNA in cells is ribosomal RNA. So maybe I'll write something like 80% of RNA is the ribosomal one.
Yeah.
Yeah, and ribosomes themselves are constructed from several rRNAs and like dozens of proteins. And half-life, it could be measured in days or weeks, so they can gradually accumulate. So like here's an example of a eukaryotic, mammalian ribosome. So we have essentially 49. So for the larger subunit, right, we have 49 ribosomal proteins, plus some 5S ribosomal RNA, plus 5.8S ribosomal RNA, and you see how the ribosomal proteins are essentially, you just treat them as proteins, so there are a bunch of them. And the ribosomal RNAs, you see how there's double-stranded regions, right?
Mm-hmm.
So that's why they have, these types of, remember, like, complex three-dimensional shapes. Also, the addition of a 28S ribosomal RNA, so that's that. And they all combine to make this 60S subunit for the ribosome. And then for the smaller subunit, it's 33 ribosomal proteins plus 18S ribosomal RNA, which creates that 40S subunit. So this is like the large, like, cap or whatever it's called for the ribosome. What is it called? There's like, the large subunit. I guess it's just large and small. But yeah, the large subunit and the small subunit, and then they combine to make the 80S ribosome. Yeah, so like a eukaryotic ribosome, for instance, is like essentially 82, like roughly 82 different proteins plus 4 different ribosomal RNA molecules. That is what the ribosome is composed of.
Got it.
Yeah. Okay. Let's see. Processing of mammalian ribosomal RNA. So we have, RNA polymerase 3 here, RNA polymerase 1, and these. So these like circles are the polymerases, and these like blue kind of strips are essentially the ribosomal RNAs. So here's RNA polymerase 3, and it's making this 5S ribosomal RNA, and and the rest of these. So essentially what's happening here is it's just showing you how we don't need to know like it step by step, but it's just showing you how the different polymerases make different things, I suppose. So, and then it says SNO RNAs, which are small nucleolar RNAs. They help process the pre rRNA, and they attach the rRNA while being transcribed.
Okay.
Yeah, and now the rRNA in a large subunit of a prokaryotic ribosome. So we don't have to, like, yeah, this is specifically for a prokaryotic ribosome, but for a eukaryotic one, it's going to be like similar in that it's just showing us that a ribosome is a combination of ribosomal RNA and protein. And you can
Okay.
kind of see it in this image here where essentially you have, these like yellow kind of bits. These yellow bits are proteins, I believe, and then I think they're proteins because they have like the alpha helices and the beta pleated sheets, and then the gray area, I believe, are just the ribosomal RNA. Because for prokaryotes, at least, the ribosome is 2/3 rRNA and 1/3 protein. So it's going to be more ribosomal RNA than it is protein. So maybe I would just write here. I'll write something here. I would say I would write something like ribosome equals, like, maybe I'll say like whatever, prokaryotic one is essentially 2/3 ribosomal RNA plus 1/3 protein. So, yeah. So, yeah, just the important thing to remember is just that a ribosome is composed of mostly ribosomal RNA and some proteins. All right?
Mm-hmm.
Now, the nucleolus is the thing that's inside of the nucleus, and it essentially just is where ribosomes are made. So, in interphase, in the cells that are in interphase, rDNA, which is ribosomal DNA, is clustered together in the nucleolus, and most of the nucleolus is just a bunch of ribosomal subunits. In HeLa cells, around 7,000 ribosomes are produced in the nucleolus per minute.
Okay. Oh, GFP is a ribosomal protein.
Wait, what did you say about GFP?
Oh no, I said I didn't know it was a ribosomal protein.
Ribosome? Wait, what's up?
No, I just said I didn't know it was a ribosomal protein.
Ribosomal protein?
Yeah.
Which...
It says GFP right there on the slide.
Oh, yeah. So, yeah. That's just them attaching that to, ribosomal proteins to show that in the nucleolus or whatever, there is a bunch of ribosomes essentially being produced.
Got it.
Yeah. Okay. Now, transcription in eukaryotic cells. Now we're talking about transfer RNAs. So, transfer RNA. So see how it has this like clover kind of shape?
Yeah.
I kind of drew it like up, like up here as well.
Yeah.
Yeah. So let's see. Plants and animals have about 50 different transfer RNAs. Transfer DNA is repeated. There's about 500 genes in humans. The repeats are clustered in the genome. They're about 80 nucleotides long. They're transcribed using Rna Polymerase 3. So over here, I wrote like Rna Polymerase 1 transcribes rRNA, and 3 transcribes the tRNA.
Yeah.
And half-life is in days or weeks, so they gradually accumulate. So essentially, we're seeing that the half-lives of half-life in days or weeks. So the ribosomal RNA and the transfer RNA, they're going to have much longer half-life. So they get to essentially stick around. They don't get broken down like messenger RNA does. So that means that they accumulate over, the course, like they gradually accumulate over time.
Oh, okay.
Yeah, and then messenger RNAs. So that is the one that, encodes a particular polypeptide, and there's a significant non-coding segment, and that can essentially, act as they can have regulatory functions, because they can be like, an enhancer or something like that. And so here's like, a good example, like a good one for the human beta globin mRNA, which is, one of the subunits of hemoglobin.
Mm-hmm.
So essentially we have, this is the, this over here is the coding region, and it codes for polypeptide, 146 amino acids long. And then here, so these are the untranslated regions, so they don't get translated.
Mm-hmm.
So there's one at the 5 end, and there's one at the 3 end. And then attached to that will be the 5 cap and the poly A tail. I'll be right back. I'm just going to use the bathroom. Okay.
Alright, it's 1 second. Alright, I'm ready.
Yep.
Alright. Alright. Ready?
Yeah.
Well,
Are you ready?
yeah.
Okay. So we have prokaryotic mRNA versus eukaryotic mRNA. So prokaryotic mRNA doesn't need to have any type of 5 cap or poly A tail added to it, and
Mm-hmm.
it's polycistronic, which means that it contains multiple messages just from a single mRNA sequence. So, so for prokaryotic mRNA, one mRNA can can be translated into several different proteins. because it has several different genes on it. Whereas eukaryotic mRNA, each mRNA just codes for one gene, so it will just turn into one protein instead of multiple proteins. So that's monocistronic.
Okay. Wait, can you explain that one more time?
Yeah. So basically, in a prokaryote versus a eukaryote, right?
Mm-hmm.
The prokaryote will have just an mRNA translated. So maybe I can do it like this: mRNA. 5-end, 3-end. So I could say like gene 1, gene 2, gene 3, and it's translated into protein 1, whoops, protein 2, protein 3, and this happens, like all of this happens in the cytoplasm, and this is polycistronic, which means that one mRNA can code for more than one protein / gene. Okay, and I kind of wrote it out like this.
Mm-hmm.
So, for a prokaryote, their mRNA, there's no pre-mRNA or anything like that. It's just like the one mRNA that it makes, which can have multiple genes in it, and it's translated like just from that one mRNA. It can be translated into all of the proteins, all the different proteins, and all of that happens in the cytoplasm. And it's polycistronic, which means that one mRNA can code for more than one protein or gene.
Okay.
Now, for eukaryotes, you're going to have the pre-mRNA, right? Which is going to go through like alternative splicing to get rid of the introns. And it's going to add the 5 cap and the poly A tail to make the mature mRNA, and all of that happens in the nucleus. And here I'll say, like, this is the mRNA here. So like Pre Mrna gets rid of the introns, adds the 5 cap and the poly A tail to make to make the mRNA, and that all happens in the nucleus, and then it leaves the nucleus where it gets translated into just a single protein, and that's in the cytoplasm.
Okay. Slightly confusing.
Which part?
I guess this is the prokaryotic one more?
Prokaryotic one? Yeah. So the prokaryotic one can have multiple genes on a single mRNA. And then that can be translated, like every single gene on that mRNA can be translated. So multiple, that's why it's polycistronic, which means that there's multiple messages on a single messenger RNA.
Mm-hmm.
So like I have like gene 1, gene 2, gene 3. So they have like, protein alpha, protein beta, protein gamma. So there's multiple coding sequences in a prokaryotic mRNA. So like an example of that would be like the Lac Operon, right?
Mm-hmm. What does that say inside that box where the 5 and 3 are? In the prokaryotic?
Oh, sorry. So like gene 1, gene 2, gene 3?
Oh, okay.
Yeah.
And then the cytoplasm's right there.
Yeah, so that means that all of this happens in the cytoplasm because prokaryotes don't have a nucleus, right?
Mm-hmm.
Yeah.
Prokaryotes, no nucleus.
Yeah, because they don't have any membrane-bound organelles.
Yeah.
So they only have ribosomes, essentially.
Got it. And then polycistronic. One mRNA can code for greater than one protein and gene. Okay. Now it makes more sense.
Mm-hmm.
And now for the eukaryotes.
Yeah, and the eukaryotes are monocistronic, so each mRNA will code only for one gene. That'll just make one protein.
Was that still 5-3 on gene 1 there? On the third one.
Here?
On the third arrow there.
Oh
Yeah.
yeah, it's just that there's a so just like here they have like a 5' end and 3' end.
Yeah.
Here it's a 5 on the 5' end. There's a 5 cap. So that's what I drew
Okay.
here. So the 5' cap, and then yeah, I can write the 3' and stuff.
I think that was probably what was confusing me. Sorry.
Mhm.
And then that's a poly A tail. Yes, I.
Yeah. So like, for example, here I have some notes here. So this is an example of the Lac Operon. You remember that, right?
Mm-hmm.
So that's that's a perfect example of prokaryotic transcription. And so essentially, this is the mRNA for it, and see how there's Lac Z, Lac Y, and Lac A. Each of these are separate genes. Lac Z makes beta galactosidase, Lac Y makes permease, Lac A makes transacetylase, but
Mm-hmm.
it's just one mRNA here that makes all three of these proteins.
Mm-hmm.
So that's an example of a polycistronic, mRNA for prokaryote, right?
Yeah.
All right. Now and then down here I have RNA for eukaryote. So essentially, I didn't draw like the part where you get rid of the introns, but yeah, this is the 5 cap and the poly A tail, and they basically keep it from, because if you think about it, the 5' end is going to have 3 like phosphates attached to it,
Mm-hmm.
and those are negatively charged, and they can be degraded by exonucleases or like phosphatases. So the 5 cap basically protects it from it being degraded. And the 5 cap is essentially just it's guanine, like like this is like a guanosine, like like nucleotide, and then this is a like a pentose whatever sugar, and then 3 whatever phosphates. Yeah, it binds to that end, and then poly A tail is essentially just a bunch of adenines. So it's polyadenylated, but I can go more in depth on that if they talk about it more.
Okay.
Yeah, so like the main takeaway here is just that for prokaryotes, there is no pre-mRNA. There's no like alternative splicing. There's no 5 cap. There's no poly A tail, and there can be multiple genes on a single mRNA, and all of those can be translated. So it can make every single, it can make multiple proteins. But in eukaryotes, each mRNA codes for one protein.
Yeah. Hi, yeah. What's up?
Nothing. I was checking on to see how you're doing.
I'm doing okay. I'm currently with admin.
Oh, you're in your group.
Yeah.
All right, bye.
Alright, bye-bye. Alright,
All right.
back again.
So machinery used in mRNA transcription. So Rna Polymerase 2. GTF just means general transcription factors, just things that can help transcription essentially happen. And then the promoter is that special sequence of DNA. That's 24 to 32 nucleotides upstream to the transcription coding region.
Mm-hmm.
Yeah. And
Okay.
there's something called a Tata box, and it's this it's a consensus sequence, which means that it's going to be there for a lot of different types of genes. Like it's conserved, evolutionarily conserved. And yeah, it's going to be like upstream, about 25 nucleotides upstream of that start point.
Okay.
Yeah. And so that essentially so that's going to be the site of the pre-initiation complex, where the transcription factors in RNA polymerase 2 can kind of like get together and start like or start their binding process to get ready to transcribe once it gets to this start point over here. Yeah, and you'll kind of see what they mean with a pre-initiation complex, because this RNA like kind of folds in a kind of weird kind of pattern, not pattern, like into like a weird shape. So it'll start to make sense, possibly later if they show pictures. This is a Tata binding protein. So we have DNA here, and this is the, I suppose, protein that binds to it. So the pre-initiation complex is this weird looking thing here. It's really like this gigantic thing over here. Let's see if I can find another pre-initiation complex. So yeah, here's like a kind of another picture of it.
Okay.
So like, yeah. So essentially, like, you know, the promoter, essentially where the RNA polymerase binds, and you know, gene A, let's say that's the one that's going to be transcribed, and then the transcription factors are binding to it as well, and this thing essentially bends over to produce this gigantic weird thing, which is called the transcription pre-initiation complex. So that's what that is. So that's what, so like essentially, so here we see the Tata box, right? It's upstream of the start of transcription, and then this orange thing is the gene over here.
Mm-hmm.
And then we have this binding protein, transcription binding protein, and transcription factor 2D. they come into play and they bind to the Tata box, right? And then this is transcription factor 2b. There's just different types of transcription factors that also binds to this this green one over here. And then there's going to be a bunch of other things like transcription factor 2e, transcription factor 2h, transcription factor 2f. So they're all going to bind to this RNA polymerase 2. So when they eventually go to this to bind to the promoter and stuff, it's going to make this gigantic complex of a bunch of different transcription factors, polymerase 2, etc. And then it causes this, so it basically makes this weird pre-initiation complex, is what it's called. So here's another kind of picture of it. Yeah. So that's the pre-initiation complex. Now, there are many GTFs, which is just general transcription factor in this complex. The two important ones are, transcription factor 2D, which is this, the green one, because it contains TBP, which I think is probably binding protein, transcription binding protein perhaps. And then TF2H over here, this pink one. And that contains a helicase and a kinase. And then and then there's a tail which is going to be where the C-terminal domain is. So yeah, so just the main takeaway here is just that all of these things, like the Tata box, so essentially all of these things combine to make a pre-initiation complex, which is this weird, crap over here.
Okay.
So that's
should I write down from this?
So let's see. Maybe you can write something down let's see. Pre-initiation complex formed by RNA polymerase 2 GTFs, which are general transcription factors, so different types of transcription factors, and the promoter. And so what the pre-initiation complex is a bunch of GTFs that bind to RNA polymerase 2 at the promoter and Tata box to produce a, let's say, three-dimensional protein complex that is the machinery for transcription. So, let me zoom out a bit. So, this will make more sense, I suppose, as we go down to the slides and you see that, you see more about it, but, essentially, it's a complex of approximately 100 proteins that's necessary for the transcription of protein-coding genes in eukaryotes.
Okay.
Yeah. So it's a bunch of this stuff happening. So, various different transcription factors. So I just wrote. So the pre-initiation complex is formed by RNA polymerase 2, GTFs, and the promoter. And what it is essentially it's a bunch of GTFs that bind to RNA polymerase 2 at the promoter and Tata box to produce a 3D protein complex that's the machinery for transcription. This picture is pretty good. So it's essentially just this gigantic protein-like complex made of different proteins that happens before transcription is initiated. Okay?
Okay.
Does that make sense?
Yeah.
All right. Yeah, so it's this thing right here. Okay. Now, initiation of transcription. Let's see. The C-terminal... Alright, the C-terminal domain, CTD, of RNA polymerase 2, of an RNA polymerase 2 subunit is phosphorylated by transcription factor 2H. Phosphorylation releases polymerase from the promoter site, and transcription factor 2D remains bound to Tata to initiate additional transcription.
Okay.
Yeah, so I'll write some stuff about that because it's talking about. So it says that these are the two most important GTFs. So I'll write most important GTFs are let's see, TF2 H and so what that does is it phosphorylates the C-terminal domain of an RNA polymerase 2 subunit, and what that does is it releases the polymerase from the promoter site. Oh, here, let me do it so you can see. Okay. And then TF2D. So what that does is it remains bound to Tata to initiate additional transcription.
C-terminal is what it is?
Yeah. Like you know, proteins will have the N-terminal, C-terminal, right?
Mm-hmm.
Yeah, and then I wrote like the overall function of the pre-initiation complex in blue over here. So essentially what it does is it positions the RNA polymerase 2 at the gene transcription start sites in addition to denaturing the DNA, which, makes the double strand, separate. And it positions the DNA in the RNA polymerase 2 active site for transcription. So that's the overall function of the pre-initiation complex.
Denatures the DNA, that says?
Yeah. Which, means that you take the double strand and you spread it, you separate the strands.
Mm-hmm. Okay.
Okay.
Not to rush, but we
Mhm.
kind of got to start picking up the pace, even though we're meeting again this
Yeah.
week and stuff. We still have another PowerPoint that has like 38 slides. So
Yeah.
in order to make sure we don't fall behind, we got to pick up the pace just a tiny bit.
Okay. Let's see. Okay, so here's just a picture that shows, a transcription unit. So there's a promoter, right? And that's what the RNA
Okay.
polymerase binds to. Then it, transcribes, the gene. And then there's a terminator, or the termination point, where it'll stop.
Mm-hmm.
All right, now...
Yeah, if you go down to like the fourth one, it has the picture of the entire thing.
Okay.
There you go.
All right, and then, so okay, that's the pre-initiation, and now once the polymerase is attached, it's the initiation. So this is the unwound DNA, the RNA transcript that's being made, and then, so that's just the start, and then elongation is when you're just adding more DNTPs to that growing RNA transcript. And, you see how the polymerase is moving to the right?
Mm-hmm.
You see that, right?
Yeah.
Okay. And, and then once the polymerase reaches the end, the terminator, right, it causes termination. So then the polymerase gets released from the DNA, and the completed RNA transcript is released. So that's what that is, okay?
Mm-hmm.
And then this is an overview of the processing of pre-mRNA. So there's hnRNA, which is heterogeneous nuclear RNA. So that's just a pre-RNA that's processed to mRNA. So it's the same three things that, happen. Essentially, 5-cap is added, 3 poly A tail is added, and then, the alternative splicing, or the removal of introns.
Okay.
Alright, now this is what the 5-cap and the poly A tail are. So, essentially, on the 5 end, there's going to be 3 phosphates there. So, it removes the phosphate and it adds a methylated guanosine. And what that does is it protects the 5 end from exonucleases. So nuclease is just, you know, an enzyme that breaks apart like a nucleic acid. And if it's exo, it means that it breaks it apart at the ends. If it's endonuclease, it breaks it apart in the middle. Or somewhere in the middle, right? Not at the ends. And the 5' also aids in the transport from the nucleus, and it helps initiate translation. Now, the poly A tail is on the 3' and so an endonuclease forms a new 3' by recognizing this AAU-AAA sequence. So this means that a polymerase adds about 200 to 250 adenosines. It doesn't need a template for that. And what that does is it protects it from premature degradation, and it also helps initiate translation.
Got it.
All right, so these two things are, you know, added to the ends of the Pre Mrna to turn it into the mRNA. All right. Now, eukaryotic and bacterial genes are organized differently. So okay, for a bacterial gene, we just have something like this. Doesn't have any introns or anything like that. You know, and the green thing is the promoter. But for eukaryotic gene, you know, there's a bunch of exons, a bunch of introns, so there's going to be alternative splicing, which allows for genetic recombination between the exons and drives gene evolution, because there's going to be lots of different protein isoforms that will form. Alright, now RNA splicing of split genes, which is removal of introns. So, you know, we already talked about that. Here's just an example for an adenovirus, I suppose. So, you know, yeah, we have like, these are the introns, and then there's a hexon gene, and, you know, this is the primary transcript, and then they process it, so this is the intermediate here, so they add, like, you know, poly A tail and stuff, and then they get rid of these introns to give you, you know, that. Alright. Nucleotide sequences at the splice sites of Pre Mrna. So, yeah. So, these are just particular sequences that are required for the removal of introns. So, like this one, I guess it's GGU, and that just indicates that that it's going to start to remove it at that area. And then this, you know, pink underline here is another sequence at the splice site, and it just lets you know, it lets it know that it's going to cut those out and splice them together at the exons.
Okay.
Yeah. Okay, now this is the role of SN Rnps and spliceosomes in mRNA splicing. So, yeah, so this is another example of, you know, mRNA acting as an enzyme. Because it basically, like the splicing, it carries out itself. It doesn't, like, require another enzyme. And so, you know, so basically what it does, so we have the mRNA here with the intron, and so basically we have a combination of proteins and SN Rnps. So what those are small nuclear riboproteins. All right.
Okay.
So that's essentially a combination of protein and SN RNA. And then some other proteins will help make this thing called a spliceosome. And the spliceosome is the thing that causes the alternative splicing. And what's important to see here is just that it's not caused by a separate enzyme. It's caused by the RNA itself acting as an enzyme. So the spliceosome is a complex containing a variety of proteins and small nuclear riboproteins. There's consensus splice sites, aka these things, like that area and that area right there. At the 5' end and the 3' end, and they base pair with the Rnps. So this over here and this are the consensus splice sites, and they interact as in, they form base pairs with Rnps. So you see how they're connecting to Rnps here?
Yeah.
Like right there and here.
Mm-hmm.
And so a lariat is just like the shape that it makes. I think it means like a loop or something like that. And it just causes this intron to be spliced out. So that gets spliced out. So this is the excised intron, which means that it's, cut out. And then the spliceosome components leave, and you're left with the mRNA that's been spliced, so that intron leaves. So you have the mRNA.
I see, okay.
Yeah. Now, so here's some more information on RNA splicing. I guess it means identification of various SNRNPs, and actual steps are not important for this class. Good. So you just know the general idea of how that works. And then let's see, this is a possible mechanism for coordination of pre-mRNA processing. So let's see, we have 5 capping. Okay, this is just talking more about what we've already talked about. Which means like it gets spliced, it gets a 5 cap, a poly A tail to make, mRNA. All right, now how the cell can tell that mRNA is mature? So a mature and intact mRNA will have a 5 cap, poly A tail, and has been spliced. Binding proteins recognize these modifications and then move the mRNA into the cytoplasm. So here we have the 5 cap, exon junction complex, and poly A binding protein. The poly A tail. And this green thing is a poly A binding protein. This orange one is the cap binding protein. So those are the binding proteins that recognize the modifications, right? To see if it's a mature mRNA, and if it has those things, it lets it exit the nucleus through the nuclear pore, and then it enters the cytoplasm, and then these binding proteins leave, and then it creates that initiation complex, and then it gets translated, right?
Mm-hmm.
All right. Alternative splicing, we went over that before, so this can be spliced in different orders and stuff like that, right?
Yeah.
Let's see, correspondence between exons and protein domains. Okay. All right, that's just talking about, this exon over here is responsible for this domain, this one is responsible for that domain, etc. Translation is RNA turning into protein, so this step. Now, properties of the genetic code. So codons are triplets of nucleotides, so like 3 nucleotides are a codon. They're non-overlapping. So, here's like UGG, that's a codon. And it codes for tryptophan over there. So,
Okay.
Yeah.
So, it's 5:56, which basically means we're out of time.
Yeah.
And it's probably going to open up into something that needs much more explaining.
Okay.
So we should start with that on Friday.
Oh, start with what?
This on Friday.
With this? Like where we left off here?
Yeah, where we left off.
Okay.
Right here. Yeah. Slide 56.
All right. Yeah, and yeah, let me know, like, you know, because I don't know, like, if you're like writing things down or whatever. So let me know, like, how to adjust the pace.
Oh yeah, no, you can always send over your notes and stuff, but I'm writing down most of it just in case I miss anything. You can always send it over.
Yeah, I mean, just let me know about the pacing.
Oh, I'm doing okay with pacing.
Okay.
Okay, yeah.
All right. Okay, so like, wait, when was your test again?
October 4th.
October 4th. Okay, so that's it's like 1.5 weeks.
Yeah.
Okay, and so next time we can like, yeah, I suppose we can finish where we left off, and then we can look at the practice test.
Yeah.
Yeah, I took a look at.
Remember, I go in now to take it.
Oh, like in person?
Yeah, we're not doing it online.
Oh, okay. Yeah, so I mean, I took a look at the practice test. It didn't look too bad. So we can like, yeah, next time we can take a look at that, okay?
Okay.
Yeah, it doesn't look too bad from what I see. All right. Okay, so
Alright.
that's it, right? All right.
Yeah. So I'll see you Friday.
All right, man. All right, take care.
All right. Take care.
All right.